Hi, my name is Yulan van Oppen, and I'll be presenting fast parameter estimation for ODE-based models of heterogeneous populations. This is my main PhD project that I've been working on with my supervisor Andreas Milias. Simply put, this research is about efficiently incorporating variability in parameter estimation and considering distributions of parameters often more appropriate than fixed point estimation when cell measurements deviate a lot. The current gold standard for inferring parameter distributions is the global two-stage or GTS method, which can be very accurate but relies on non-convex optimization. A large class of biomechanical models express non-linearity with respect to the states, but linearity when it comes to the parameters in terms of their differential equation. So our contribution is an alternative implementation of the GTS method that leverages this feature through gradient matching. Which is additionally able to handle hidden states with an EM algorithm. The models of interest are nonlinear mixed effects models, and here we focus on those that are ODE-based. These models assume each cell is governed by a differential equation of the same form f, but with a different parameter factor beta I. These parameter factors are referred to as random effects and are assumed to be distributed with mean B and covariance matrix D. The mean B is called the vector of fixed effects. And the data are assumed to be noisy samples from the cell-specific ODE solutions. The GTS approach first estimates each cell-specific parameter vector, then combines the information about the estimate spread and their uncertainty Ci to infer the random effects distribution. The GTS method's first stage optimizes cell-specific parameters using trajectory matching. This is a form of nonlinear regression that minimizes discrepancy between the data and the numerically integrated solution given a parameter vector. But the iterative optimization schemes require numerous repeated numerical integrations, amounting to a considerable need for computational resources. An alternative approach is gradient matching, where the regression takes place on smoothing-based estimates of the gradients in the data instead. What's nice is that smoothing data is generally a lightweight task and no numerical solutions are required at any point. So let's get to our contribution. We have adapted the GTS method to use gradient matching in its first stage instead of trajectory matching, where we adopted the common practice of smoothing data using B-splines, hence the name gradient matching GTS or GMGTS. Of particular interest are systems which are linear in parameters, for which we can form a linear regression model. So we can use efficient convex optimization approaches like a generalized least squares estimation. And although it's omitted from the poster, the challenge becomes efficiently approximating the first stage estimates uncertainty Ci for the second stage. We demonstrate our method's performance using a fluorescent protein maturation example. The system happens to be also linear in its states, but that's certainly not required. Only linearity in parameters exploited. Figure 4 shows some sample data as circles, B-spline smoothing as dotted lines, and the fitted trajectories as dashed lines. We compare the two implementations in terms of accuracy and computing time. Figure 5 shows the inferred joint distribution of KDP and KM, as well as the ground truth, to visualize the small loss in accuracy for the GMGTS method. However, as we can see in table 1, the computation time is over 30 times shorter for 100 simulated cell trajectories, even when mRNA and dark protein are hidden. You may have noticed the linear regression system requires full state information, but in practice such data are rare, so we designed an EM algorithm to deal with this problem. So after smoothing the observed state and given an initial guess or a fit to the population average, we integrate the system to obtain the estimated hidden state information. Together with our gradients, we can reconstruct the linear system and perform gradient matching. And these two steps are iterated until convergence. Estimating the corresponding uncertainty Ci is a bit more involved, but we have derived an efficient approximation that requires only the system's parameter sensitivity and the solution to another linear system. So although numerical integrations aren't completely avoided this way, orders of magnitude fewer are needed compared to the direct nonlinear optimization. So, to summarize, we can get a substantial computational gain through gradient matching using linear regression, while still handling partial observations. And also, no parameter initializations are required when all states are observed. Nevertheless, there is a small loss in accuracy to take into account, and not unimportantly, high quality smoothing is crucial for accurate solutions. We're currently looking into making this step interactive, so that visual assessment of the smoothing quality and tuning of the hyperparameters can be simultaneous. All estimations and plots were generated in MATLAB using the IQM tools and Monolix software packages. And if you're interested in some background material on mixed effects models, the GTS method, or gradient matching, you can check out the top 3 references. Thank you for watching.